Alright guys, I'm going to pick up right here where Kyle left off. Obviously he wasn't smart enough to do this section about molecular biology here. Just kidding, he's sitting right here next to me. He's one of the smartest guys I know. There's going to be 9 questions on this section. Let's jump right into them. This first part, DNA structure and function, has the majority of the questions from what we consistently see. The first thing we want to look at is the structure of DNA. You can see right here that it has 3 parts. Let me show you how that was asked in past test questions. Guys, as you looked at the answers to those questions, make sure you remember that anything that ends in ose is always a sugar. So we have a sugar, a phosphate, and an amine base group. You guys can see there that there's purines and pyrimidines. The important thing to know is that A goes with T and G goes with C. Let's talk a little bit about bonds in between. There's hydrogen bonds between A and T. There's 2 of those between A and T. G and C always has 3, so there's a little more strength holding those together. You might see that in a question about melting temperatures, and the higher the content of G and C, the higher the melting temperature is going to be. The other thing you need to know there is that phosphodiester bond, that's a covalent bond that runs along the sugar phosphate backbone that holds it together a lot more tightly. Those hydrogen bonds aren't as strong, so when we need to unzip the DNA to translate it or replicate it, that can be done easily. Let's take a look at how they've asked some questions in the past on this. We've had several questions there on A and T and G and C going together. I want to make sure you're not confused by that last question, the molar percent question. It's simply wanting you to make sure you understand that if you have 20% of A, that corresponds with 20% of T, which is going to be 40%. That leaves 60%, so that means half of that, or 30%, is going to be G, and the other half, 30%, is going to be C. It's as simple as that. The next thing I want to talk about are histones. You guys remember that they bind to DNA, give it structure and support? Take a look at this test question and make sure and remember what they're highly concentrated in. The next topic we're going to hit is DNA replication. You guys know this happens within the nucleus. Let's take a look at this diagram and see how it all works. As you look at this diagram, I want to stress 4 things. We've got to unwind it, we've got to start it, we've got to build it, we've got to stitch it back together. Helicase comes in and unwinds the DNA. Primase comes in and starts the process. DNA polymerase comes in and builds the chain. Remember, this always happens in the 5' to 3' direction. So our leading strand is running downhill, and our lagging strand, this has to happen in fragments. That's where DNA ligase comes in. Let me show you a question to make sure you remember exactly what ligase does. Take a look at this heading called RNA structure and function. I know you guys know that U replaces T when we get to RNA. So A goes with U. Do you remember what nutrient is needed to form purines and pyrimidines? Take a look at this test question. Take a look at the heading nucleotide synthesis and metabolism. You're going to see a lot of enzymes listed here. I want to focus in on a couple key ones. We want to look at what it takes to synthesize and to break down these different bases. Let's take a look at a couple questions and how they've asked it. Guys, at most you're going to get 1 question on this, but make sure you circle and know those enzymes. Alright, as a reminder guys, transcription is DNA to RNA, translation is RNA to proteins. Remember, transcription happens inside the nucleus of the cell. It's got to be transcribed to mRNA to get out of the nucleus, into the cytoplasm where the ribosomes are, and that's where protein synthesis happens. So let's take a look at this diagram. I know you guys have seen translation many, many times. I just want to refresh you here. This is happening in the cytoplasm. The rRNA, or the ribosome, is where this is happening. Sometimes we'll see a question that asks about the site where protein synthesis is happening. mRNA is coming in. Remind yourself of the function of tRNA by looking at this question. Guys, you're going to see a text box about reverse transcriptase. You guys know you've seen that related to HIV. Here's a way they've asked that in a test question. The next heading we're going to hit is on DNA repair and mutations. This is a real low yield section. We haven't seen many questions on it. Read it if you have time. If not, here's a couple examples of how we've seen questions. Take a look at these and you're good to go. Guys, as we look at the cell cycle, the big thing I want you to understand is that any DNA replication, translation, or protein synthesis all happens outside of mitosis. Let me show you an example of how this has been asked on the old test questions. The next topic that's been consistently on old tests is the northern blot, southern blot, and western blot. Here's a mnemonic to remember that. Now let me show you some past test questions on how this can be asked. All right, guys, last point here. Let's look at gel electrophoresis. Here they want you to know that at the isoelectric point, a molecule will not move. You make molecules move to separate them by changing the pH. If the pH drops below the isoelectric point, you're adding protons. Remember, protons are positive, so that's going to move to the negative pole or anode. Let's look at a couple of test questions. Guys, that is it. Congratulations on being done with another big section in your Big Five. Let's move on to dominate some more.